Hello class, let's start a lecture on cell membrane biochemistry. At the end of the lesson, students should be able to describe the chemistry of bacterial cell wall, the plasma membrane, mechanisms involved in transport, and mitochondrial respiration. Chemistry of bacterial cell wall is very important because in order for us to develop drugs that can be able to target the bacterial cell wall and destroy it, we need to know what the biochemistry of bacterial cell wall entails. Plasma membrane also, what is its biochemistry, transport mechanisms, and also mitochondrial respiration. The cell envelope refers to the structure that surrounds bacterial cells, and this includes the plasma membrane, a cell wall, and in some cases a second phospholipid bilayer membrane. Let's watch a short video on the cell membrane.
Professor Dave, let's talk about the cell membrane.
He knows a lot about the science stuff, Professor Dave explains.
We probably know that living organisms are made of cells. From unicellular organisms like bacteria, which are made of just one cell, to large animals that are made of trillions of cells. One key feature of a cell is the cell membrane or plasma membrane, which separates what's inside the cell from what's outside of the cell. This is a very important function because without this separation, there could be no such thing as life, because an organism must be distinct from its surroundings. Inside a cell is where metabolism takes place and genetic information is stored under regulated conditions or homeostasis. The cell membrane is semi-permeable, meaning it lets some things through while preventing other things. And now that we know about lipids, we are ready to understand this outermost layer of the cell in more detail. That's because the structure of the plasma membrane is similar to the soap micelles we learned about, where the molecules arrange themselves with the hydrophilic heads facing out and the hydrophobic tails pointing in, except that instead of a micelle, it is a phospholipid bilayer that engulfs the contents of the cell. It's called a bilayer because it's two layers of phospholipids, which each have a phosphate group similar to the carboxylate group in the soap molecules, with formal charges that can interact with water molecules, and nonpolar fatty acid tails, one saturated, the other unsaturated, which prefer to stay hidden from water molecules. One layer faces outwards to the aqueous environment outside of the cell, and the other layer faces inwards to the aqueous environment inside the cell. That means there are two layers of nonpolar material in between. This is part of what makes this membrane semi-permeable. Some compounds are able to traverse this nonpolar section, and others are not. We refer to this bilayer as a fluid mosaic because these phospholipids are not fixed in their location with respect to one another. They are swimming around and constantly changing places, like concert goers navigating through the crowd. But it's not just phospholipids in the membrane. There are molecules of cholesterol, which give the cell membrane some rigidity. Otherwise, it would be too fluid. In addition, the cell membrane is filled with many different proteins that have a wide variety of functions. Some of these are channel proteins, which float about in the sea of phospholipids and facilitate the movement of some kind of molecule across the membrane. Let's learn about these proteins and the different ways that substances can move in or out of the cell. Some molecules don't need help to get through the membrane. Small nonpolar molecules are able to go right through the lipid bilayer because they are not repelled by this nonpolar material. This is how oxygen and carbon dioxide get across, since they are tiny and nonpolar. This process is an example of diffusion, where molecules move across the membrane along with a concentration gases spontaneously fill up a room. Other molecules need help to get in and out of the cell. Some molecules can get through via passive transport, which doesn't cost any energy. This includes polar molecules like water and glucose, as well as various ions which can move through special transmembrane proteins that connect the intracellular space and extracellular space, so that these polar or formally charged particles don't have to push their way through the non-polar tails of the lipid bilayer. This kind of transport doesn't require energy expenditure when the particles are moving with the concentration gradient, because it is an entropically favorable process, and we can refer to this process as facilitated diffusion. So something like a sodium or potassium ion, when moving with the gradient from high concentration to low concentration, can pass through by passive transport, which is a kind of diffusion. But when things need to move against the concentration gradient, active transport is required. Since this is an entropically unfavorable process, there will have to be an energy expenditure involved. So certain proteins, like a sodium-potassium pump, use up ATP, the molecule that is the currency of cellular energy, to move sodium ions out of the cell and potassium ions into the cell against the concentration gradient, from low concentration to high concentration. We will talk more about ATP later. Transport proteins that assist in either passive or active transport are highly specific, each allowing only a particular substance or small group of similar substances to pass through. Aquaporins move water. Ion channels move ions, and so forth. Some of these are channel proteins that remain open, and some are carrier proteins, which alternate between two conformations, like the glucose transporter, where binding with glucose causes it to change shape, and then, once glucose is released, it changes back to its original conformation. Beyond transport proteins, there are many other types of proteins in the cell membrane. There are glycoproteins with oligosaccharide chains jutting out that help cells recognize one another. There are scaffold proteins that bind with structures inside or outside of the cell to maintain cell shape and location. And lastly, there are receptor proteins that receive signals from outside of the cell. Let's learn about those next. Thanks for watching, guys. Subscribe to my channel for more tutorials, and as always, feel...
Okay, so the cell membrane of a bacteria, so we have the gram-negative and gram-positive bacteria. These are the two classifications we have for bacteria. We have gram-negative bacteria and gram-positive. So, the bacterial envelope or cell envelope comprises of, we have the gram-negative and the gram-positive bacteria. So, over here, the gram-positive bacteria has lipoteichoic acid. That's the structure here. It also has teichoic acid. This is the structure. But its peptidoglycan layer is very thick. It extends from here to here. Whereas that of the gram-negative extends from here to here. That's the peptidoglycan layer of the gram-negative. It's very thin. And the gram-negative organisms don't also have teichoic acid, but they have porine or aquaporine through which water and other molecules pass through. So this is the peptidoglycan layer. It is very thin, whereas that of the gram-positive one is very thick. It has teichoic acid, gram-positive. And in terms of pathogenicity, or how serious their infection is, the gram-negative organisms are most of the disease-causing bacteria we see in the human host, while the gram-positive is not so serious. The gram-positive, most of them are not serious infections. So, an overview of the cell envelope. We have the gram-positive bacteria. You can see the yellow rectangular boxes represent the peptidoglycan chain or peptidoglycan layer. It's very thick, and this is the plasma membrane with integral proteins and phospholipid bilayer. We can also see that the gram-negative bacteria have both an outer cell membrane and an inner cell membrane with a very thin peptidoglycan layer. You can see it's very thin as compared to the gram-positive bacteria. The bacterial cell wall consists of peptidoglycan, which is an essential protective barrier for bacterial cells that encapsulates the cytoplasmic membrane of both gram-positive and gram-negative bacteria. So, when you'll be doing as you progress in your career, you'll be doing gram staining to identify which organisms are gram-negative and which organisms are gram-positive. Gram-negative organisms are able to retain the primary stain. The gram-negatives, they are able to retain the primary stain because of the thick peptidoglycan layer, while the gram-positive positive organisms are able to retain the gram stain. That is, the initial gram stain is it crystal violet or so. Then the last one is crystal violet? I've forgotten the stain. So it's able to retain the primary stain, whereas the gram-negative bacteria has a very thin peptidoglycan layer, so it's not able to retain the primary stain. So it loses the primary stain and rather absorbs the secondary the secondary stain, or the secondary stain, which is neutral red or saffronin or so. So the thickness of the peptidoglycan layer makes it possible for it to retain the initial stain that we use to stain the bacteria during gram staining, while the thinness of the gram-negative bacteria makes it not able to retain the primary stain or keep the primary stain, but it loses the primary stain and rather absorbs a secondary stain. So the bacterial cell wall consists of peptidoglycan, which is an essential protective barrier for bacterial cells that encapsulates the cytoplasmic membrane of both gram-positive and gram-negative bacteria. The peptidoglycan is a polymer consisting of sugars and amino acids that forms a mesh-like layer outside plasma membrane of most bacterial cells. So it comprises of N-acetylglucosamine and N-acetylmuramic acid. And these two sugar units are alternating and they are joined together. The N-acetylmuramic acid has a peptide chain of 3 to 5 amino acids attached to it. So you see, we have N-acetylmuramic acid and N-acetylglucosamine. These are the alternating chains found in the peptidoglycan layer. So you can see, this is just a monomer of the peptidoglycan layer. And the N-acetylmuramic acid has a pentapeptide, or 5 amino acids attached to it. We have the L-alanine, D-glutamine, L-lysine, D-alanine, D-alanine. So the N-acetylglucosamine and the N-acetylmuramic acid forms a meshwork like this, with the adjoining tetrapeptide chain, which is bonded to the N-acetylmuramic acid. So you can see the tetrapeptide amino acids. 4 amino acids is called tetrapeptide. Then there is an interbridge of peptide or amino acids also here. then another 4 tetrapeptide chain joining still the N-acetylmuramic acid. So you can see the chain. It is a very rigid protective barrier for the bacteria. So this is also an overview of the peptidoglycan chain. So if I should ask you what are the components or the biochemistry of the peptidoglycan chain, you should be able to say N-acetylglucosamine, N-acetylmuramic acid forms an interconnecting chain which is bonded to by 3 to 5 amino acids. Then you name the amino acids either L-alanine, dextrorotatory glutamine, levorotatory lysine, dextrorotatory alanine. Importance of the peptidoglycan chain. Peptidoglycan serves as a structural role in the bacterial cell wall. It counteracts the osmotic pressure of the cytoplasm. It's also involved in binary fission during bacterial cell reproduction. So let's look at what binary fission is. So if you have a cell like this, for reproducing by binary, this is nucleus. When it's reproducing by binary fission, it produces what? 2 daughter cells, which are of the same size as it. 2 daughter cells. And the 2 daughter cells, they are all of the same size as the parent or the mother cell. Each 2 daughter cell will also divide. So upon division, we have 4 daughter cells. And these 4 daughter cells will also divide. And this process is called mitosis. Mitosis. Let's proceed. So, let's proceed. So, yes, so that's binary fission. Also, is also termed mitosis. Okay, so gram negative. You can see the gram positive, the thick, this blue thick peptidoglycan layer. Then you can see the gram negative is very thin peptidoglycan layer, very thin. The gram negative also have 2 plasma membranes, one inner plasma membrane and one outer plasma membrane. Properties of cell membrane. We finish the selective permeability. The cell membrane is selectively permeable. This is because it allows some but not all materials to cross into or out of the cell. It is a very biased cell membrane. It enables cell to maintain homeostasis. Homeostasis means what? Internal constant internal physiological environment. For instance, if you want to maintain homeostasis, let's say you have COVID-19, you have developed COVID-19, or you have been infected with COVID-19. So, your temperature will spike. So the normal physiological temperature we have here is 35.5 degrees C to 37.8 or 0.9 degrees C. If you have COVID infection, then your temperature will now rise to 38 and beyond. Meaning that some of the enzymes in your body are going to be affected. They will start burning, they will start dying. So your hypothalamus will now is a part of your brain responsible for regulation of temperature and other things, sleep and other things. So it will tell your body that no, your temperature is so high, so produce more sweat. Produce sweat. Produce more sweat. So that there will be what? Cooling. So the cooling effect will reduce the temperature back to normal. So restoring the temperature back to normal. When we produce more sweat, we reduce the temperature. We restore the temperature to normal or reduce the temperature. Then we take in a lot of water. Apart from taking a lot of water, you also have to you will not be urinating frequently, going to toilet frequently. That is how the hypothalamus will program the body. So homeostasis is restored. A constant internal environment, homeostasis will be installed or restored. Constant internal environment, constant or normal temperature of the body, normal physiological temperature will be restored. So this is the structure of the plasma membrane. We have the lipid bilayer, the phospholipid, the phosphate group, and the lipid group. We have integral or transmembrane proteins. They span through the plasma membrane. They run through the plasma membrane. Then the integral or peripheral membrane proteins, they are just attached to the membrane, the outer side of the membrane. They don't span through or go through the membrane like this. So these big proteins that span the membrane are transmembrane proteins, and they form the ion channels, the aquaporins, and the protein transporters, because they span the membrane. We also have oligosaccharides, which is a sugar. We also have glycoproteins, this is a protein saccharide that has been joined to a transmembrane protein, so it becomes what? A glycoprotein. We have the phospholipid and the bilayer, etc. So this is the inside the cell, that's the cytoplasmic side, and this is the extracellular side. This is your assignment. Lipid peroxidation in plasma membrane. This is a very serious phenomenon. Lipid peroxidation. This is free radical generation. For instance, if you have water like this, and the water is broken down to release hydrogen proton plus what? OH. This is normal thing that is supposed to be done. But when there is radiation, or you eat some foods, or the sunlight on any other thing, some chemicals can trigger cellular processes, abnormal cellular processes, so that the H2O will now be broken down to what? H plus OH dot, and the dot is an electron. With the release of a free electron, so we end up, this electron is unpaired. Over here, this electron means what? Dot, dot, 2 electrons. But now it is only one. One has been lost over here due to radiation, UV light, and other things. So you end up, this is a free radical, very dangerous. It's a rebel. It will go, we call it free radical. Free radical. It's a radical student. It will go and destroy tissues, cells, and even cause permanent mutation of DNA. Permanent single nucleotide mutation, and SMP, single nucleotide mutation, and destroy cells, destroy plasma membrane, single nucleotide mutations. So this is the result of cancers, skin cancer, colorectal cancer, and a whole lot of problems we are facing on this earth is due to free radicals. Another one is like this: lipid peroxidation. Once if you have butanoic acid, butanoic acid is 4 carbons. Butanoic acid, 4 carbons. Look at butanoic acid. It's supposed to have a COOH. Now, what happens is that let me replace this by R, this long thing by R. That's the alkyl group. So when it's broken down, instead of it having this is minus plus H plus. But when there's a loss of electron, E minus, then this one will now lead to what? Formation of a free radical. This is what we call lipid peroxidation. It will lose one electron here. This electron, one will be lost. So that we will have only one electron. And this one is an unstable. It's very unstable. This electron is very unstable. This compound is very unstable. Because you know, the women who are not married, they want to have boyfriends. So they will go and snatch somebody's husband. So by way of snatching somebody's husband, they will go and tell the man that, man, buy me this, pay my rent, pay my this, pay my this, because they are not stable, they are not married, so they have only one electron here. They will go and destroy marriage people's home, destroy their marriage, cause broken home, because they are having only one electron, so they think they don't have money. So they have to go and snatch people's husbands. And this is not good. Free radicals can cause wear and tear of muscles. Free radicals are not good. They are the result of all the problems on the earth. And they cause cancer. You are visiting a friend with your mother. You get to your friend's house, and your friend will give you a chair and will not give your mother a chair. The meaning of that is that your face looks like an old person. Why? Because you have a lot of free radicals in you. You are chewing a lot of fried rice, KFC, and other things. So it's causing lipid peroxidation, breakdown of lipids. Saturated fatty acids are being broken down, and we don't have and they are not. It ends up producing free radicals, which will go and destroy cells. So the abnormal destruction of DNA cells, plasma membrane, leads to cancer and also leads to growth. You see that some people, they are about 45 years or 50 years, but they look like somebody who is 16 years. Others too are young, but they look very old. If you are young and you look very old, it means you have a lot of free radicals. So you have to start taking antioxidants. And some of the antioxidants that we have, an example is what vitamin C. Vitamin C is an antioxidant. Vitamin C is an antioxidant. Vitamin C is an antioxidant. It's called ascorbic acid. Ascorbic acid. So that's why when COVID-19 came, we are advised to be taking soluble, we are advised to take vitamin C. Vitamin E. Vitamin E is also an antioxidant called tocopherol. It is called tocopherol. Tocopherol. Called tocopherol. They are all free natural antioxidants. An antioxidant, what an antioxidant does is that it is against free radicals, antioxidant. It is against free radicals, and it fights against what? Free radicals. So antioxidant vitamin C will come and donate one electron, and it will donate an electron to the hydroxyl free radical, and to produce OH. And the OH means 2 electrons. This dash means 2 electrons. So this electron will go and join to this electron to produce this. And this one, because it is it is now stable, because it has to be 2 electrons but not 1. It will stop going about chasing other people's husbands, chasing other people's wives, sugar mummies, because it is now married. This is marriage. Let's go back to the slide. So the lipid peroxidation producing these free radicals will end up destroying the cell membrane. The yellow we see is the cholesterol. Cholesterol is part of cell membrane. That is what the cell membrane, you can see the yellow inside. The cell membrane itself is a fluid mosaic model. Fluid mosaic. It means that fluid mosaic model. It means that the cell membrane flows like a water body. It flows like a liquid. It flows like a liquid. So the cholesterol there is supposed to stabilize it or make it rigid. Make it rigid so that it will not flow like water too much, but it will flow like water, but it will attach some form of sense to it. It will regulate the flow so it will not be too much, and the flowing will not be too much. That's the work of the cholesterol there. So when there is lipid peroxidation, there is what? Free radicals like hydroxyl free radicals, superoxide, anion, they all go to cause, this is superoxide. Is it O3 or so? It goes to cause wear and tear. Superoxide, and I forgot so they go and cause destruction of cell membranes, DNA, and the rest. Even they will go and cause destruction of the mitochondria, Golgi apparatus. So this is a normal cell. When the cell is attacked by free radicals, we also have the hydride free radical. Hydride. This one, only 1 electron here. It becomes hydride. Hydride. HY. Hydride free radical. It's very dangerous. If it is like this, it's a hydride anion. But if it's 1 electron has disappeared, it's hydride. It will go and destroy cells. So you can see that this normal cell has been attacked under oxidative stress. When there is too much of free radicals in your body, we call it oxidative stress. So look at the destruction. So it will lead to cellular necrosis. The necrosis, another name for necrosis is what? Unprogrammed cell death. Necrosis. Necrosis is called unprogrammed or unnatural cell death. The cell will die unnatural, premature. And it's not natural cell death. But if it's apoptosis is defined as apoptosis is what? Programmed cell death. That is, the white blood cells or the red blood cells have been programmed to have a lifespan of 120 days. So programmed cell death. So they are programmed cell death. They are supposed to die. They are programmed to die by 120 days. Neutrophils are programmed to die, or white blood cells, some white blood cells called neutrophils. They are programmed to die within 3 days. And they mediate inflammation reactions. So neutrophils are supposed to die within 3 days. So programmed cell death is called apoptosis. Unprogrammed cell death or premature cell death is called what? Necrosis. Okay, so transport processes. We will be looking at permeability, passive and active transport, transport mechanisms. Only small uncharged molecules, such as gases, water, ammonia, glycerol, or urea, are able to pass through biological membranes by free diffusion. Examples, you can see free diffusion, simple diffusion. Small molecules like oxygen molecule, nitrogen molecule, benzene can just traverse, easily pass through the cell membrane. Then polar uncharged molecule. These are molecules bearing no charge and they are of a smaller molecular weight. They can pass through the cell membrane easily. With increasing size, when the size of the molecule is so big, even compounds of this type that are no longer able are not bearing charges are no longer able to pass through the cell membrane. Examples are glucose and other sugars like mannose, fructose, and the rest. You see, glucose under normal circumstances is supposed to pass through the cell membrane because it has no charge. But because it is a big molecule, it cannot pass through. The same way ions cannot pass through the cell membrane because they are charged. And you know the phospholipid bilayer is negatively charged. This blue thing is negatively charged, both inside and outside. And the outside of the cell is aqueous. The inside, the yellow is the fatty acid or lipid. It is what? Uncharged. It is lipid. But the outside is polar, the cytoplasm is polar. So you cannot pass through. You cannot use water to pass through kerosene or water to mix with what? Oil or fried oil. It is not possible. So the pharmaceutical companies, what they do is that if they encounter something like this, what they do is that they will develop something we call liposome, so that it will liposome to transport liposome. So if you have an active ingredient which is polar, something like if you have an active ingredient as sodium, whatever, CO as an active ingredient in a drug. This is just an example I'm giving. If you have something like this as an active ingredient in a drug, and it is charged like this, you cannot pass through to the cell membrane, so you have to put it in a liposome. A liposome is like this. It's a fat, it's a container which is made up of what? Fat. So you put this thing over here in the middle. When you put it in the middle, then this fatty environment or liposome will now pass it through the cell membrane, because the liposome is fat, and the cell membrane is also what? Fatty. So the liposome can pass through to go and deliver this drug to the cytoplasm. So let's look at how these ions can be transported. This glucose can be transported. So we see permeability. Hydrophobic molecules, like molecules that hate the molecules that are afraid of lipid or fat, they cannot pass through. They can molecules that are these molecules can pass through the cell membrane because they have no charge and they are smaller size. These molecules are also able to pass through the cell membrane, ethanol, glycerol, urea, because they are smaller molecular weight. But glucose is big, sucrose is big, it cannot go through. Sodium, potassium, ions, calcium, they have charges, so they cannot go through the cell membrane. To be able to take up or release molecules that are strongly polar compounds, particularly those that are electrically charged, cells depend on specialized channels and transporters in their membranes. Meaning that for us to be able to traverse or transport or be able to diffuse polar substances, charged substances, or big molecular weight substances through the cell membrane, the cells use what? Protein transporters. So we'll be seeing them. So let's look at free diffusion. It's the simplest form of membrane transport. When it is supported by integral membrane proteins, it is known as facilitated diffusion or facilitated transport. Let's look at it. So we have some channels in the cell membrane. Examples are what? Ion channels and porins. So if it's difficult, if the particle that we are sending through the cell membrane is very big and it's difficult to traverse the cell membrane, then we have to use ion channels or porins. So let's look at them. So we have what? Ion channels. Ion channels, mind you, they transport only ions. So we have sodium ions, calcium ions, potassium ions, chloride ions. They pass through ion channels. Then porins are pores. They are pores. Pores. A pore is a hole. They are pores, which can allow passage of what? Smaller molecules to pass through or bigger molecules to pass through. And aquaporins are what? Aquaporins. Aqua means water. Allow only water to pass through. So aquaporins allow water to pass through them. So this is the plasma membrane. We have what? A porin channel or a pore, which allows particles to pass through, bigger particles. Transporters recognize and bind the molecules to be transported and help them to pass through the membrane as a result of conformational change. So you see, this is the extracellular milieu or extracellular environment, and this is the intracellular space or the cytoplasm is here. So you can see the substances you want to, or the molecules you want to transport across the membrane. This protein receptor or a protein transporter will have a conformational, it has a strong affinity or strong attraction for these molecules. So when these molecules bind onto it, it will now go through a conformational change. It will change its shape so that it can be able to accommodate and transport the particle across the cell membrane. So it will change its shape. You can see that it has opened its mouth to deliver the particle, then it will now change back to this shape. So now it has changed its shape. It has not opened the mouth. It is rather pregnant. Now it will release the content. So it has to open its anus. At first the mouth was open. Now it has opened its anus to deliver. So now it took in food. The food entered the belly. It came out of the anus. Now it has to open its mouth for another food. So direction of transport proteins. So we have, if we transport substances across, when we transport substances across transmembrane protein, only one type of molecule, we call it a uniport. But if there are two different molecules, like sodium and magnesium, passing through the same protein transporter, but in the same direction, we say it is a symport transport. It is a symport transport. If they are antiparallel or in different directions, we say antiport. So features of transporters. Transporters are proteins. So all these transporters you see, they are proteins. Integral proteins or transmembrane proteins, the same. They are very specific to molecules they transport. Yes, this one will transport maybe sodium ion. It will not go and transport potassium or anything. It will not go and transport sugar. They are specific. A sugar transporter will transport only sugar. Amino acid transporter will transport only amino acid. Maximum transport capacity. They have a high transport capacity. They transport a lot of particles through them. They have greater affinity or greater attraction to the molecules they transport. Free diffusion and transport processes are influenced by concentration gradient. So free diffusion and transport processes facilitated by ion channels and transport proteins, always follow a concentration gradient. The direction of transport is from the site of higher concentration to the site of lower concentration. We all know free diffusion. So for free diffusion, we see that the ions are low here and the ions are very high here. So if you are moving using this arrow, the arrow means that the ions are more here and they are flowing here until equilibrium is sustained. This is what we call free diffusion or free diffusion. But if the ions are passing through this protein transporter or pouring, if they are passing through these ion channels, against a concentration gradient, this is a highly concentrated place and low concentrated place, and they are passing through against this concentration gradient, we say it's active transport. Let me explain active transport. Okay, free diffusion. Free diffusion. Diffusion. Hey, how do you spell diffusion? Let me spell like this. Diffusion. Okay. So if I buy a spray, I have a spray, a bottle of spray. Like this. And I press the canister and the spray fills the whole room. The whole room. This is the barrier or the gate of the room or the boundaries of the room. So I stand outside the door and spray. And after spraying, the spray travels here and enters here. And fills this whole room. And fills the room. So that equilibrium is attained. Equal amount of spray over here will be the same as spray over here. We say that there is what? Equilibrium or free diffusion. Okay, what about if I have some molecules over here, 5 of them, and I have a barrier over here? I have a barrier over here, and these molecules are able to move, and the same molecules are here, but there are plenty here. There are plenty molecules are here, and few are here, and these few molecules are moving from here to here, because normally if there is molecules are concentrated here, the spray is concentrated here, and you spray, it's supposed to pass through this membrane, semi-permeable membrane, and go and fill up here, and until we have equal amount of spray particles here and equal amount of spray particles here. So if we have 20 spray particles here, then over here across the membrane is also supposed to be 20. So if you have 20 spray particles, 50 spray particles here, and they move, 25 will remain here and 25 will remain here. By an active transport, it is some way. Active transport is some way. It's like a greedy wife. You know you are a greedy wife. It's not good. Because see, your husband's salary is 250 Ghana cedis, and you are taking a salary of what? 2,500 Ghana cedis. Then you expect this man to give you chop money. After giving you chop money, you expect him to use the 2.5 million to pay school fees, and also to buy you clothes, funeral clothes, so that you can attend funeral, to buy you lipstick and other things. You are a wicked woman. That is active transport. The man does not have money. The man does not have money, and is using all this money, the 2.5 million, to take care of you, whilst you are earning 2,500, 25 million. You are a wicked woman. So that is the explanation for active transport, meaning that the particles will move from a region of lower concentration to a region of higher concentration. So here the man, at the end of every month, the man will be left with what? 0 Ghana cedis. This is cheating, wickedness. And the woman will be left with what? 2,500 plus 250 will be what? 2,750 Ghana cedi. What kind of greedy woman is this? So this is active transport. And active transport requires the expenditure of energy in the form of what? ATP. So it has to use ATP, convert ATP from ATP to what? ADP. So ATP will now be converted to what? ADP, to release what? Energy. Then the ADP will also converted to what? AMP. Or we can remove a pyrophosphate, that's 2 phosphate groups, to produce AMP. So the man, meaning that for him to be able to spend the 250 Ghana cedi on the wife and on the children, he needs to do extra work. He needs to do overtime at the work site. He needs to work so hard. He doesn't need to sleep. He needs to borrow money, loans and other things to be able to satisfy the wife and the children. So he needs to expend a lot of energy, go through a lot of stress to be able to pass these 250 Ghana cedi to the wife. So these are the particles. Small salary, representing the particles. Then the woman will be relaxing and be receiving it. This is the explanation for active transport. We need energy to pass these small molecules across this semi-permeable membrane to the other side, where there is a lot of molecules of the particles of the substance we are transporting. So let's see. So you see, for low concentration or gradient of the ion, we have what? Protons. Small number of protons, and they are being transported across or through the protein receptors, through the protein transporters to the other side of the cell membrane, where the protons are concentrated. Then, for a good wife, she will say that, oh, my husband, your salary is small. Let me spend the 2,500 on you, because your salary is small. So when the good wife is spending her money on the husband, that becomes what? Simple diffusion or free diffusion. There is no expenditure of energy. But with the active transport, there is energy requirement. So you can see examples of facilitated diffusion. This is the facilitated diffusion. When we are diffusing substances from a region of higher concentration to a region of lower concentration of the particles, it's called diffusion. If it's passing through the plasma membrane freely, we call it was simple diffusion or free diffusion. But if it's passing through protein transporters, we call it what? Facilitated diffusion. So you can see here we have four particles outside, then one inside. And under normal circumstance for diffusion, the particles are supposed to flow from a region of higher concentration of the particles to a region of lower concentration until there is equilibration or equilibrium is attained. 3 particles here, 3 particles here, equal, so there will not be net movement either inside or outside. The same, we have 5 particles here, 1 inside. It will flow until we have 3 here and 3 here. So there is a balance. We are all earning the same salary, so nobody is going to spend on anybody. For the active transport, like I explained, it requires ATP expenditure, and we move particles from a region of lower concentration to a region of higher concentration. You need a lot of energy to overcome the concentration gradient barrier. So this is what a husband earning 250 Ghana cedi and the wife earning 2,500 cedi. But the wife wants the husband to spend the money on her. So the husband had to work so hard to be able to spend the money on the wife. So passive transport, you can see Free diffusion goes through the cells, but facilitated goes through protein transporters. Then active transport, you see, we started with what? Outside we have only 2 of these square boxes, 2, and inside we have a lot of the square boxes. We have 5. So we move these extra 2 square boxes to go and add to the greedy wife who already has 5, but we need to expend ATP or energy. Let's pause here and continue.